Good morning, good evening, everyone. Thanks for attending the webinar. My name is Yue, and I am the Technical Application Liaison from Mission Bio. And today, I am going to introduce you to our single-cell sequencing platform, Tapestry, and how our multi-omics analysis can help advance precision medicine. So this is just a brief overview of today's agenda. First, I'd like to discuss about current challenges of bulk sequencing and how Tapestry is unique in the industry. And second, I'd like to give you a technical overview of the platform. And lastly, but not least, is I'm going to talk about what is the impact of measuring multi-analytes, including genotype and immunophenotype, at the same time in the same single cell. Here, I'd like to provide some brief background about Mission Bio. So we are a late-stage startup in a rapid growth with more than 100 instruments worldwide. Since launching the first instrument back in December 2017, till today, we have more than 30 peer-reviewed publications, and over half of the NCI Cancer Institute are using our technology. Worldwide-wise, we have over 800 end users offering both catalog and custom panels, both for DNA and protein. Our platform offers an integrated DNA and protein analysis, and our team has the expertise in microfluidics, molecular biology, and bioinformatics. So as many of the recent research and publications suggest, cancer is actually a highly heterogeneous disease, and most cancer, they actually harbors many different clones. And as showing on this graph, these clones are related but not identical, and their identity is usually defined by the different mutational profile that they harbor inside of the cell, as well as immunophenotype based on their cell surface marker. And depending on the genetic makeup of these clones, and they could actually grow differently, respond to therapies differently. So bulk sequencing is a great technology to study cancer genetics. However, it does have a few limitations to understand, especially the intra-tumor heterogeneity due to its population averaged approach. You often only get the VAF value or variant of allele frequency representing the population average for each of the mutation that you identify from that sample. And you can only infer rather than measure the actual mutational frequency, especially in single cell level. Plus, once you have immune cells, stroma cells, other normal cell that is contaminating your tumor sample, it would actually confound your mutational profile result. Nevertheless, to mention, using bulk sequencing data, it is difficult to measure the actual zygosity, co-occurrence pattern if you detect multiple mutations in that same sample and detect rare clones. And last but not least, using bulk sequencing data, you often just get one single analyte, which is the mutational profile of that sample, but you are missing the cell surface protein markers, which is the immunophenotype of that cells. So it's very, as you can tell, that they are none of the, so all of them are very important in order to differentiate different cell type, different clones, to detect intra as well as inter-tumor heterogeneity. And in the scenario that if you have a clinical application here with very limited primary material, you often have to split one sample across multiple platforms. And these data can sometimes be very hard to integrate and would require a high amount of starting materials, which is usually hard to get in a clinical setting. So here, this is where Tapestry comes in with a novel multi-parametric assay within a single workflow that both conserves the sample and consolidates multi-omics assays into an integrated single-cell resolution dataset with very easy-to-use analysis software. So this is the workflow. Tapestry is a high-throughput droplet microfluidic system that sits upstream of standard Illumina sequencing. It works by enabling encapsulating single cells into droplets. Within these droplets, we do a highly high-plexed targeted amplification directly of the genomic DNA. During this amplification of targeted loci, the cell identifying barcode is integrated into each amplicon as part of the triple primer PCR system. And to generate the protein detection data, you can just add on to a brief antibody staining protocol, deploying a catalog panel or a custom antibody oligoconjugate cocktail. And this barcode of antibody will be subsequently digested during the first encapsulation protocol. However, it will leave behind the oligo barcode within that droplet. And this oligo is then captured and integrated into the genotype library and reported as corresponding NGS reads after sequencing. And this is how you can associate, the DNA sequencing data as well as the cell surface protein sequencing data in the same cell, single cell, by sharing the same cell barcode. And we also recently validated a low input protocol to address the previously inaccessible samples due to low starting cell put. So this is a recent launch of our low input protocol. So on the right bar graph below, we see a consistent single cell DNA sequencing performance observed across a range of cell inputs from 20,000 to 100,000 cells, as demonstrated on the left-hand side by the constant capture rate more than 5% and a high panel uniformity of more than 90%, showing on this right across the range. In terms of what kind of input material is compatible with our platform, there's actually a variety of them. First, if you're just working with DNA, you can either fresh or frozen, even just non-viable samples, they are completely compatible with our platform because we are working with DNA here. We also work with sample that is cryopreserved in DMSOs or single cell suspension, such as PBMCs, BMMCs, and some of our researchers can even do a upfront FACS or MAX enrichment of certain cell population if they're interested in specifically for single cell sequencing. And we can even work with, dissociated fresh frozen tissues. And, like some customer, they work with mouse models, doing a xenograft mouse models. We can also work with that. And some people working with, doing cell line research. Those are like no-brainer for compatibility with our platform. And in terms of the cell input, since we have validated the protocol, we can go down to 20,000 single cells without compromising our cell capture rate. So currently our range of input from 20,000 all the way to 100,000 cells. And the capture rate is about 5 to 10%. So up to 10,000 cells will come out of the 100,000 cell, 20,000 to 100,000 cell input. And in terms of the result that you can obtain for the DNA side, on the DNA side, you can get the the SNVs, copy number changes, small indels deletions, and even internal tandem duplications, structural variants through our special white glove design. And you can also simultaneously detect the cell surface protein expression profile using the panel that we have launched with BioLegend, or build your own custom panel with BioLegend. And in addition to the easy-to-use instrumentation and fully kitted reagents, we provide a suite of software solutions to support upfront panel selection, custom design, as well as an integrated multi-omics pipeline for multi-omics analysis. And finally, you can use Tapestry Insights for data visualization and generate publication-ready figures. And in terms of the panel that we offer, currently we offer 3 catalog panels plus 3 publication panels in the hematology panel side. We also have these virtual panels that can be your inspiration resources that you can build these panels based on your own needs. We also have one catalog panel in the solid tumor side, which is a tumor hotspot panel, a pan-cancer panel, basically. And you can also use, in terms of solid tumor, we have this universal nuclei isolation protocol that you can use as input. And you can also build your own custom panel here by working with our design scientist for your own purposes. For example, if you are doing genome editing or use mouse models. In terms of the application that Mission Bio offers, two major applications here. So if you're just measuring, for example, DNA using our single cell DNA sequencing, our applications enable the researchers to map clone evolution, clonal selection, and look at the clonal trajectory. For example, the researcher can determine a rare clinical relevant subclone population, identify driver clone responsible for drug resistance, resolve the mutation zygosities, copy numbers, and how these mutations are being acquired in orders. And with our multi-omics technology, researchers can further differentiate the cell population by both use the genotypes and immunophenotype information here. The researchers can identify the cell type and simultaneously to look at the specific genotype that are being harbored within each cell type. And the potential clinical utility of our instrument includes minimal residue disease detection, differentiating benign somatic mosaicism from active neoplasia, and even our technology has been widely used in genes and cell therapy product characterization, or doing for those companies who are doing gene editing approaches optimization. So this graph truly shows how our instrument has evolved and evolved through, incorporating a single analyte from using DNA sequencing to a multi-analyte approach. Comparing to, the traditional bulk sequencing data, you can further differentiate your cell population into different clones based on the mutational profile, different mutation, how they co-occur in each other in these different clones. And further, you could actually differentiate your cell populations by either their SNV profile or CNV profile. And by incorporating the cell surface protein expression profile further, you could look at the protein expression, cell surface protein expression profile within each of the genotype clone, identify the clones, and trying to, or on the vice versa, you could differentiate different cell types, and then look back what kind of mutation is being harbored in these different cell types. And further, this multi-analyte approach allows you to differentiate the different cell population into a better resolution. As you can see there, that if you're differentiate for different cell lines based on just the SNV or CNV, none of them are ideal. Neither of them are ideal. However, if you're incorporating 3 different analytes together, these 4 different cell lines can be perfectly separating from each other. And last but not least, I just want to give you an example of the multi-analyte analysis approach that we have done in a primary AML sample. So we have collaborated with Dr. Chris Horrigan from NIH, and he provided a primary AML patient sample who have known to have, chromosome 4, chromosome 7 copy number loss. And then we have run tapestry on both their DNA and protein, use a 45 amplicon panel and 6 antibody panel. And by looking at the SNV or CNV, as you can see, they can differentiate the cancer and normal population, but using the protein, you can even further differentiate the cancer population into a major cancer and a sub-cancer population. And the immune cell, you can also determine different cell population based on their cell surface protein markers, CD3 and CD19. So after you detect, you have run these on the cell population, you can also overlap the 3 analytes in the same heat map, and you can see that they correspond to each other perfectly. First, you see that in this experiment that we have run around 4,000 cells, and in the major cell mixture cancer and subcancer population, that's differentiated by not just by cell surface protein markers, but also by the mutational profile. For example, ASXL1, which is a pathogenic mutation that is actually appearing as a heterozygous mutation in the cancer population, but it's all wild type in the healthy cell population. Plus, you can also determine the copy number loss of the chromosome 4 and chromosome 7 in the cancer cell population. As well as this can be validated using a SNP profile, because you can see that the SNP is showing or as a heterozygous mutation in the healthy cell population. However, in the cancer cell population group, you see a change of status, zygosity status from heterozygous mutation into wild type. So they corresponding perfectly to each other. On top of the genotype information you can obtain, you can also obtain the different cell types within in the cancer cell population and outside of the cancer population. And that's all for today.